Welcome to the Experiment 01, Basic Optical Systems. In this experiment you will work with some of the most important tools in optics: lenses, polarizers, photodetectors, and a monochromator. These are devices you will frequently encounter in research laboratories. The goal of these demonstration videos is to help you prepare before entering the lab. Please note, these videos are for demonstration purposes only. When performing the experiments, you must follow the detailed instructions in the lab manual, along with all course guidelines and safety rules. Your lab notebooks and reports should reflect your own work in the lab, not these demonstrations. In every experiment, your first priority is safety. Always observe the safety instructions provided. When you arrive in the lab, the equipment will already be set up for you. Before you leave, make sure everything is returned to its initial state. Handle optical components with care, and avoid touching the glass surfaces at any time. Careful setup and observation will make your work more efficient, accurate, and rewarding. We will explore 3 topics in 01. You'll use lenses to form images, test the thin lens equation, and set up a compound microscope. You'll rotate polarizers to generate and analyze linearly polarized light, and test the Malus law. You'll scan different wavelengths through a monochromator and record how the photodiode responds. Check the optical components in the box. There is a viewing screen in a bag used to view real images, a diaphragm or iris that controls the amount of light passing through, a diffuser that causes light to spread evenly and removes bright spots, a crossed arrow target that serves as the object in the imaging system, 3 convex lenses of different focal lengths, 3 polarizers, a photodetector, and a 630 nanometer long-pass filter. Check the optical bench. The light source is already mounted, which you can switch on. Several holders are provided for mounting optical components. To align optics properly along the bench, press the holder base against the rail. Each holder has a white line to help you read its position on the meter scale. Remember, there is an offset between the center of optical components and this white marker, which you need to correct for. Set up an image forming system with the tungsten lamp, a diffuser, a crossed arrow target, a viewing screen, and a lens. Adjust the viewing screen back and forth until a sharp image appears. At this point, you can measure the object distance, DO, and the image distance, DI. If the focal length of the lens is unknown, you can use the thin lens equation to calculate it. Notice that when DO is very large, DI approaches F. This means a quick way to estimate the focal length is to image a faraway object and measure the image distance. With a lens of known focal length, you can now verify the lens equation and explore different imaging situations. Change the object distance, refocus the screen each time, and record how the image size, orientation, and position change. To test the lens equation, you can plot DI versus DO, which should trace out a hyperbola. A better approach is to plot 1 over DI against 1 over DO. The data should fall on a straight line with a slope of -1 and an intercept of 1 over F. Next, set up a compound microscope using 2 lenses. Use the 75mm focal length lens as the objective to form a real image of the crossed arrow target. Start with the objective placed about twice its focal length away from the target, so that the image distance is also close to twice the focal length. Now add the eyepiece, a 127mm focal length lens. Position it so that the real image from the objective lies within the focal length of the eyepiece. In this way, the eyepiece produces a magnified virtual image at your near point. Look through the eyepiece and slowly adjust its position until you see a clear, sharp image of the arrow target. To increase the magnification, move the objective lens closer to the target. As the position of the real image shifts, you'll need to adjust the eyepiece to keep the image in focus. You can also move the objective in the XY plane perpendicular to the rail, to examine different regions of the target. In addition, you can explore ways to control optical aberrations, that is, the distortions or blurring in the image caused by the breakdown of the paraxial approximation. Let's now turn to another fundamental property of light, polarization. Begin by mounting a polarizer on a holder. Each holder has a white marker that allows you to align and read out the axis of the polarizer. Set up the polarizer in front of the tungsten lamp, projecting the light onto a viewing screen. For a clearer view, you can also use a lens to image the lamp onto the screen. The polarizer only allows light with a specific polarization direction to pass through. By rotating the polarizer, we can examine different polarization components of the light. Unpolarized light is made up of waves vibrating in all directions, with no single preferred orientation. The light emerging from the first polarizer is linearly polarized. Now add a second polarizer to test this polarization. As you rotate the second polarizer, you project the electric field onto different axes, so the transmitted intensity changes with the angle between the two polarizers. Next, use a photodiode to record how the transmitted intensity depends on the angle, and test Malus' law quantitatively. Mount the photodiode securely with a clamp, and connect it to a multimeter by matching the same polarity or the same colored leads together. Carefully align the photodiode in the X, Y, and Z directions, and adjust its tilt so that as much light as possible falls on the active area. Set the multimeter to DC mode, measure voltage, and select an appropriate range that matches the signal intensity. To verify the Malus law, you can plot the intensity as a function of relative angle theta, either in Cartesian or polar coordinates, and fit it to the cosine squared function. Or you can plot the intensity directly against cosine theta squared, so that all the data points fall on a straight line with zero intercept. Lastly, let's examine how a semiconductor photodetector responds to light of different energies. We'll use a tungsten lamp together with a monochromator. Switch on the lamp and open the shutter. The lamp produces white light, which contains a broad spectrum of wavelengths. To isolate a particular wavelength, we pass the light through the monochromator. The monochromator is a 4F optical system. The entrance slit, a concave mirror, the grating, the second concave mirror, and the exit slit are all separated by one focal length of the concave mirror. If the grating were replaced with a mirror, the light from the entrance slit would go through two Fourier transforms and form an image directly at the exit slit. With the grating in place, different wavelengths are diffracted into different angles, and the exit slit selects one wavelength to pass through. Turn the knob on the monochromator and watch as different colors of light appear at the exit slit, covering the visible spectrum from about 400 to 800 nanometers. As you do this, the grating inside is rotating, sending different wavelengths into the fixed exit slit. If you continue dialing to longer wavelengths, you'll begin to see the second-order diffraction of the visible spectrum, extending from 800 up to about 1,600 nanometers. This behavior is explained by the grating equation, in which the product m lambda determines the diffraction angle theta m, where m is the diffraction order and lambda is the wavelength. To measure the response spectrum of the photodiode, focus the light from the monochromator onto the photodiode and record the signal with a multimeter. In the near-infrared region, second-order diffraction can introduce artifacts. To remove these, place a long-pass filter in front of the monochromator to block the shorter wavelength components. Keep in mind that the spectrum you obtain is not just the intrinsic response of the photodiode, but also convolves the lamp spectrum and the response of optical components. Still, the sharp drop in photocurrent below the semiconductor bandgap clearly reveals the quantum nature of light.